All right, this lecture is going to be on muscles. So I just did a dry run of this one and it did not record. So let's try this again. So with our muscular system, we have three types of muscle: skeletal muscle, cardiac muscle, and smooth muscle. Your skeletal muscle is striated in appearance, which means it's striped. If you look at these pictures here, you can see the light and dark bands, and it's under voluntary control, meaning you have conscious awareness of when it is contracting. Your cardiac muscle is also striated in its appearance. It's found in the heart wall, but it is under involuntary control. So if you wake up in the morning and not real clear on what you're doing, the good news is your heart is going to remember to contract for you. Smooth muscle is found in hollow organ walls, places like the uterus and stomach. It's non-striated in appearance, which is why it's called smooth. It's also under involuntary control. So when you ate your lunch today, your stomach knew it needed to contract and digest that. That wasn't something you had to stop and think about happening. So the functions of our muscles: movement. They're going to pull on things to allow you to move. They're going to help hold things in position. That may be holding your arm out to the side, or it can be helping to hold other things in your body in place. They will control organ volume. You've got sphincter muscles that are little ring-shaped muscles that will act like a drawstring on some of your organs. This will help control the volume of the contents. They will move substances in the body. Cardiac muscle is going to push blood throughout the body. Peristalsis is the wave-like muscle contraction that your digestive system does to move things. They'll also generate heat. It's a byproduct of metabolism. So when you're cold, you will shiver because your body's trying to get your muscles to contract and generate some heat. The more you use the muscles, the warmer they get, the more heat you've produced. When we look at muscle movement, we can categorize some different relationships here. A prime mover is going to be the muscle that generates the majority of force. If I'm getting ready to push something out in front of my chest, in front of me, the pectoralis major muscle is going to be the prime mover. The synergists are going to be muscles that are going to support or assist the prime mover. So my deltoid muscles in my shoulders would help to hold my arms in place so that my pectoralis major muscle could be an efficient prime mover. When we talk about the agonist and antagonist relationship, the agonist is the muscle that's going to cause a movement, while an antagonist will oppose it. If we're going to bend at the elbow, doing some elbow flexion, the biceps is going to be the agonist. The triceps would be the antagonist that would oppose that motion. When we look at the muscle tissue, we have fascia that's going to help form groups of muscle. It's sometimes referred to as the deep fascia. It will hold groups of muscle together throughout the body. Your muscle fibers are your muscle cells. These are arranged parallel to each other, and we're going to have membranes that are going to surround each of these. In muscles, the term myo means muscle. Epimysium is going to be the connective tissue that's going to surround an entire muscle. Epi means on. Peri means around. When we look at your muscles, there's going to be little bundles of muscle fibers called fascicles. These fascicles will be surrounded by perimysium. And then the individual muscle fibers are going to be wrapped with endomysium. So all of these membranes are going to wrap around the muscle, and the membranes are actually going to continue beyond the muscle and then just attach to themselves to make the tendons. And then this membrane will also continue on and separate and wrap around the bone and become periosteum. So this is one continuous tissue that's going to change names depending on whether it's sitting on muscle, whether it's sitting on itself, or sitting on bone. But this is going to allow the muscles to be attached very strongly to bone. A lot of times, if a muscle is going to tear away from the bone, it isn't actually going to tear the tendon. It will actually tear off a piece of the bone with an avulsion fracture. So sarco is also a prefix that means muscle. When we look at the histology of it, the sarcolemma is going to be the plasma membrane around a muscle fiber. So muscle cells and nerve cells do tend to get their own fancy names for some of the things that we see in typical cells. The T-tubules, or transverse tubules, are going to connect the muscle fibers. These are actually continuous with the sarcolemma, and this will allow the muscle fibers next to each other to be able to communicate and share impulses. The sarcoplasm is the muscle cell cytoplasm, and the sarcoplasmic reticulum is the membranous network inside, similar to the endoplasmic reticulum. In a muscle cell, this is going to store calcium that's used for muscle contraction. This calcium being used for muscle contraction is really important, and that's why you will actually break down calcium out of your bones if needed to allow the muscles to continue to contract. The body will prioritize muscle contraction because that's going to keep your heart and lungs going, which is essential to survival. Myoglobin is a pigment similar to hemoglobin. This will carry oxygen in the muscle tissue. The myofibrils are protein filaments. These are going to run the length of the fiber, but they're not continuous. In this illustration here, you can see there's pink bands and purple bands that run lengthwise, but none of them go all the way from one end to the other. Those are your myofibrils. The thin filaments are going to be made up of actin, troponin, and tropomyosin. The thick filaments will be made up of myosin. So as these are going to overlap, it's going to form these repeating patterns that make up your sarcomeres, which are the basic functional unit of skeletal muscle. So here you would have a sarcomere, and then in this area here you would have an additional sarcomere. On the ends of the sarcomere you have the Z-discs, which are a zigzag area of dense material that's going to separate them. We have a model that we use to describe how muscles contract that uses these different zones and bands. Sometimes people find this model to be more confusing than helpful. The A band is going to be the darker area that you see here. What's going on with the A band is you've got some overlap here of myosin and actin, but you also have an area in the center where there's no overlap. This H zone in the center is lighter colored. Where you have the H zone, it is myosin only. On either side, you have the A band, or the I band. The I band is lighter colored as well because it's only actin. So A band has some overlap. The H zone in the middle is myosin only. The I band on either side is actin only. And we use this to be able to talk about how when a muscle contracts, these filaments slide past each other, and we increase the area of overlap so that the I band will get smaller and the H zone will get smaller. So I will be putting in some animations in your online classroom that will show you some of these things happening that are really helpful to watch the animations. We're just reading about it or taking notes on it. A lot of times leaves it a little bit confusing. So myosin is going to be a golf club shaped protein. This is going to be one that forms a cross bridge. Actin is a helix shaped protein, so it's going to be the pink one on here. It's going to be anchored to the Z disc, and it's got myosin binding sites. So what happens is when the myosin grabs onto the actin, we call that grabbing, forming a cross bridge. Troponin and tropomyosin are going to cover the myosin binding sites on relaxed muscle. By having them covered, it's going to prevent the myosin from being able to grab on it or pull on it. So when muscles contract, we talk about it occurring by sliding filaments. You may see this referred to as the sliding filament theory. What happens is the myosin head that's on those thick filaments is going to pull on the thin filaments that are made of actin. Those thin filaments are then going to slide towards the center of the sarcomere. So the thick slides past the thin. The filament lengths do not change. What happens is the fibers slide past each other and the sarcomere shortens because you've got more overlap. So the neuromuscular junction, this is just what it sounds like. It's the nerve-muscle junction. This is where the nerve is going to tell the muscle what to do. So this picture here, you've got your motor neuron here, and then you've got a muscle fiber. The information that travels down the motor neuron is your action potential. It's an electric signal or stimulus. The motor neuron is going to be this nerve that will deliver the stimulus to the muscle. If you've taken psychology, you may have learned about how nerves communicate with each other. This is very similar how a nerve is going to communicate with the muscle. So your motor unit is going to be the single motor neuron in all of the fibers that it will communicate with or stimulate. So if we look at this example here, and you have one of these fibers come down, you can see that it will go to multiple muscle fibers. So if you need a lot of precision, you're going to have this just go to a few muscle fibers. Someplace like your hands or your lips to be able to talk, you want to have really precise movements. So you may want to just move things a tiny little bit. Other places, like in your quadricep muscles, you're going to have really large motor units. All that does is move the knee back and forth. Extends the leg back and forth at the knee. There's not a lot of precision there. So coming back and looking at this picture here, this is your synaptic cleft. So the synapse is a site of communication. It's a cleft. It actually is a physical gap here. And that's why we have to convert this to a chemical signal, because electricity does not jump over a physical gap. So that's good news. If you're sitting by the wall, the electricity is not going to jump out and get you. So what's going to happen is the electric impulse or action potential is going to come down, and it's going to trigger a chemical to be released across that cleft. So that chemical is a neurotransmitter called acetylcholine. A neurotransmitter is just a chemical that the nervous system uses to communicate. So the sodium-potassium pump is going to be present in your muscle cells, and what it's going to do is pump sodium on one side of a membrane, potassium on the other. It's going to separate these two things. Naturally, they want to be evenly distributed on both sides of the membrane. So when you keep pumping them over to one side, when you open the sodium channel, it's going to look at it as an opportunity to spread out on both sides of the membrane again. So that is going to be what's going to spread the action potential. When it does, it's going to trigger the release of calcium. That will expose the myosin binding sites and then allow the myosin to be able to grab on to the actin and shorten the muscle. So when we look at this process of the neuromuscular junction, what's going to happen is you're going to have the action potential travel down the motor neuron, and it's going to trigger the release of this acetylcholine. So the acetylcholine is going to diffuse across the synaptic cleft, and then it will activate the motor end plate and open the sodium ion channels. So it binds to the motor end plate. This is going to generate the action potential as those sodium ions are going to flow back across the membrane. So we don't want to have the muscle staying with continuous contraction. We want this signal to stop so that we can repeat this contraction again very quickly if we need to. So the enzyme acetylcholine esterase is going to break down that acetylcholine in the synaptic cleft. So if you just rapidly try and bring your fingers back and forth like you're pinching something, and you try and do that as quickly as you can, you'll notice there's a limit to how fast you can go. And eventually, things will start to fatigue in there. You can only go so fast because each time you do that, you need to have the acetylcholine broken down in the cleft. So once the nerve has delivered that message, we're going to begin the contraction cycle. So our myosin binding sites are exposed. First thing that's going to happen is we're going to split ATP. What that means is we're going to take one of these phosphates off, and we're going to transfer it onto the myosin head. When we do that, it's an energy currency. It's like it's passed its energy on to the myosin head. That's going to cause the myosin to change shape. When the myosin changes shape, it will form the cross bridge because it will attach to the actin. Once it releases that phosphate, it's going to change shape again, and it's going to pull the actin. So we say that the myosin head swivels to slide the thin fibers past the thick fibers. That pulling is the power stroke. In order for it to relax, it's going to have to bind a second ATP, and that will allow the muscle to relax. So it's all about the myosin changing shape when it's bound to ATP and when it lets go. That's what's going to do that power stroke and pulling for you. So what's allowed it to do this is when the calcium was released, it would bind the troponin and expose the myosin binding sites for this to happen. So there are a couple of pictures in your notes here that you may find helpful. I personally find the animations more helpful in visualizing this process, but if the picture works, great, use it for you. So this energy for contraction, where are we going to get this ATP from? Sometimes you'll see a muscle contraction being referred to as a twitch. When we look at the energy sources here, try not to think of it as it's going to use all of one tank and then move to the next one and use it all, and then to the next one and use it all. Rather, it will take different proportions from these different sources of phosphate or fuel, depending on how long the muscle's been contracting and the intensity of the contraction. So initially we have creatine phosphate. This is going to provide an extra source of phosphate. It'll take the phosphate off the creatine and put it onto ADP so that it can become ATP again. This is very short term, and it only provides a few ATP. If somebody is supplementing with creatine, it is not going to be helpful for an endurance activity. What this would be helpful for is maybe lifting something that's just a little bit heavier or doing another repetition, but it's not going to help somebody run a marathon faster. So that's going to be a real short-term energy supply. After about 15 seconds, you start to run low on that creatine phosphate. You're going to have to rely more on the aerobic metabolism from glycolysis. In this process, you're going to take glucose and split it in half to make pyruvate. This is going to give you a couple more ATP. This does not last a terribly long time either, only about 30 to 40 seconds depending on the intensity. So it's short term and it's relatively few ATP. You only get 2 for each glucose. So if you imagine you're going to go to a track and you're going to run full speed around, you're going to do an absolute sprint on there, what's going to happen after about a minute, you will pretty well deplete these two systems. And the legs are not going to want to continue that. So they're going to be pretty uncomfortable at that point. With aerobic respiration or metabolism, this is going to occur in the presence of oxygen. So you would take that pyruvate and you're going to keep breaking it down. It's going to be done through the Krebs cycle and electron transport chain. Those are going to give you a longer term source of energy, higher amount of ATP. However, it's just not going to give it to you as fast. And these will occur in the mitochondria. So when you're doing that sprint, you were still doing some of the aerobic respiration, but it was slowly doling out the ATP and you needed it faster. So you had to rely on the glycolysis and creatine phosphate. But once you exceeded what all of those could do, that's when the muscles were going to realize they had no more energy to contract. Now, if you slow it down to an easy jog or a walk, where you're not exceeding this system, this can be sustained for a longer period of time, possibly hours. So the more mitochondria you have, the more of this aerobic respiration you can do. Myoglobin is also important here. It's your oxygen binding molecule. The more oxygen you can have in the muscle, the more aerobic metabolism you can do. Excuse me. Lactic acid, this is going to be produced when the oxygen levels are low. So if you're having to do a lot more of that glycolysis faster than what you can do this aerobic metabolism, some of that pyruvate is going to get sent over to be lactic acid. And that byproduct will build up. So we used to think it was responsible for delayed onset muscle soreness. We know that's not the case. It does actually clear out pretty fast. It's not very pleasant feeling in your muscles. However, it's not going to be there long term. That delayed onset muscle soreness we think now is actually due to microtrauma and changes in the electrolyte balance in the muscles more so than the lactic acid. So when you have muscle fatigue, it's the inability to forcefully contract. It isn't just that you woke up that morning and you didn't want to. Chemically, you can't. So this will happen after prolonged activity. What's happened is you've had a decline in the calcium levels. There's not necessarily enough to be able to expose those myosin binding sites. The pH can change. It can become more acidic from having all of that lactic acid, depleting the creatine phosphate, insufficient oxygen to provide at the rate you need. Glycogen's depleted. You can deplete the acetylcholine so you can't even get the message to the muscle. And then accumulating the lactic acid and ADP. We used to think when you would exercise afterwards there was an oxygen debt that needed to be paid back. You had to pay back what you needed, what you had to borrow to get things started. And we realized that's not really the case. After you're done, there is an increase in temperature and metabolism that goes on. Your body temperature is actually elevated for about 5 hours after exercise. If you're an early morning exerciser, what you may notice is at lunchtime, sometimes there can really be a drop and a tendency to want to take a nap. And part of that comes from the body temperature dropping after about 5 hours, and it makes you feel a little sleepier. So for recovery, you actually need the increased oxygen after exercise for tissue repairs. The heart's still working harder. There's still an increase in body reactions. Things are still going faster. So when we look at the different types of skeletal muscle fibers, I apologize, the columns on here are shifted a little bit, but they should be okay in the version of notes that you get to download. The names give you a lot of hints. Slow oxidative fibers. Oxidative means using oxygen. Slow, going to be more of a slow, sustained type of contraction. These are going to be red fibers because they have high amounts of myoglobin to carry the oxygen because they're oxidative. They're smaller in diameter. They don't need to store a lot of fuel because they're good at making it with oxygen. But they will have a slower pace contraction. They are fatigue resistant, and they will have a lot of mitochondria to be able to do that oxidative metabolism. At the other end of the spectrum, you have the fast glycolytic. So these are going to be white fibers because they have relatively low amounts of myoglobin. They're large diameter because they need to store a lot of glycogen, but they are going to be powerful muscles. They have few mitochondria because they're not even doing that type of metabolism, but they will fatigue very quickly. Then your fast oxidative fibers are kind of the middle of the road. They're intermediate size. They're dark red because they have some myoglobin. They'll have moderate endurance, moderate resistance to fatigue, make a moderate amount of ATP, and have moderate strength. So if you've exceeded the capacity of either of these, then you have to try and rely on these fast oxidative. And they're middle of the road. They're not going to excel at either of those. So those slow oxidative are great for endurance training. The fast glycolytic, they're good for strength training. And then your middle of the road fibers here, fast oxidative, they're going to have moderate fatigue. They're just middle of the road, not excelling at either one. This picture here allows you to see light and dark colored fibers. So here where you've got the darker fibers, you have more myoglobin in here. These are also going to be smaller diameter. So when you think about a turkey and the turkey meat, in the turkey's legs, you've got the dark meat. That's going to be good for slow oxidative. When you think about what a turkey does, it walks around all day. It doesn't really run, it walks around, needs to have good endurance. In the turkey breast, you're going to have the light-colored meat. These are larger diameter fibers in here, less myoglobin, making it lighter in color. For a turkey, they don't fly long distances. A turkey doesn't migrate across the United States. However, it may need to occasionally fly over a fence to protect itself. So the reason that the dark meat has more fat, it's not that turkeys have chubby thighs, but there are more membranes, and membranes are made from phospholipid, so it's more lipid content just due to the extra membranes from the smaller fibers. When we look at muscle contraction, we can categorize it different ways. One way is to call it isometric. Iso means stays the same, metric is length. This is going to be a contraction where the length of the fiber doesn't change, but you can change how hard you're contracting. Imagine you're pushing against a cement wall. If you push lightly, the fiber is the same length as if you push hard because the wall's going nowhere. You've changed the contraction force. With isotonic, the contraction force stays the same, but the length changes. This would be more if you're using a pulley system. If you're using the pulley overhead, it's going to take the same amount of force as if you're using the pulley down lower. You've just changed the length of the muscle for your arms to be in a different position. When we look at cardiac muscle, it's going to have the intercalated discs. These thickenings in the sarcolemma will allow for the rapid conduction of the impulse. We want to have the muscle cells working together for it to be an effective pump. Muscle cells have autorhythmicity. It's a natural intrinsic rhythmic contraction in the cardiac muscle. When we get to the section on the heart, we'll talk about the pacemaker that's going to coordinate that so it actually works together instead of every muscle contracting at its own rate. For smooth muscle, we have visceral single unit and multi-unit smooth muscle. With visceral single unit, it's sheets of muscle. This is what you're going to have around blood vessels, and it will contract as a unit. This is a good thing around blood vessels because if you just had a single area of it contract, it would be like putting a piece of elastic around a balloon. It's going to bulge on either side, and that's going to be really dangerous for the blood vessels. If you have the entire sheet constrict, then you don't have a constriction point that's going to have ballooning and potential tearing. The multi-unit smooth muscle, this would be something like the erector pili muscle that goes to hair. You can have just certain hairs stand up. You don't have to have goosebumps all over your body. If you have a dog and you take a dog on a walk, one of the things that will happen a lot of times when they encounter another dog is they will have some of the hair on their back stand up. It looks like they get a mohawk along their spine. They've got individual innervation, so they can have the hair stand up on their back. It doesn't have to stand up all over the entire dog, just along their back. When we look at skeletal muscle anatomy, it's pretty universal how we describe it. The origin is the fixed point. It's where the muscle starts. The insertion is the movable point, and the action is what it does. So when we describe muscle anatomy, just about any source you look at is going to break it down into these three parts. It isn't that it's left to you to figure out which part does what. And that is the conclusion of the muscles.